Thank you very much for the nice introduction. And actually, let's see what she went to. Right here. Thank you very much for the nice introduction. So as she said, my name is Yaisa Andrews-Zwilling, and I'm going to talk to you today about recent developments in Alzheimer's disease research. So just a little bit about my background. I'm from the J. David Gladstone Institute. It's located in San Francisco, and it's an independent research organization, and we're affiliated with the University of California, San Francisco, UCSF. So the institute is divided into four different parts. So we tackle all of the major diseases that are affecting mankind currently, such as cardiovascular disease, virology and immunology, and we focus on HIV and also diabetes, neurological disease, and also translational research. Am I speaking too quickly? I get a little fast when I'm nervous. It's okay? Okay. So this guy, his name is Shinya Yamanaka, and actually this year, he's from our institute, he's a professor who works with us, and he won the Nobel Prize earlier this year, just actually a week ago, 2 weeks, for medicine, and he works on stem cells, and that's also a part of the work that I do, and I'm really proud of the institute and of Shinya for winning this. And on Thursday, it was actually declared Shinya Yamanaka Day in San Francisco for the work that was done. Yeah. So that's just a little plug for the work that we do. Okay, so a little bit about me. So I'm originally from Trinidad and Tobago, as she said. It's a really lovely island. It's 2 islands in the Caribbean. And it's a fantastic place to live and to have a childhood and grow up. And I also studied there. I went to university. But for science, which was my passion, it wasn't the best place. So I went to a place which was excellent for science, which was Germany. And this is what I faced. So it was a little bit of a shock to my system. And actually, this was my bicycle. It was a little bit tough. I didn't have a lot of money, so I had to go everywhere by bike. I biked in rain, snow, sleet, hail. But apart from that, it was a really excellent place to study. So I did my master's and PhD in neuroscience in Germany, and I also met my husband and had a kid, so it was a great program. PhD, husband, baby, I got it all. And but during the time that I studied there, every year, I was lucky enough that my parents paid for me to come back home because I was so homesick. And this is actually my grandfather. I'm not sure if you can see the picture well. And he was my biggest cheerleader while I was studying. He was so proud of me becoming a doctor. He thought I was the MD kind and not the PhD kind, but still, he was very proud. But while I was in Germany, he suffered a stroke, and he didn't recover very well from it. And after the stroke, he also started to suffer from dementia. So I actually lost him twice. I lost him because every time I went home, he could remember less and less about who I was. The last couple of times I went home, he thought I was my mother, and the last time I went, he didn't know who I was. And this was really heartbreaking for me, and actually, in some ways, it was almost a relief when he passed away, because to see him suffer like that and to see us lose the essence of who he was really heartbreaking. And this is also what kind of guided my research. Initially, I was just really interested in the brain and how it worked, but seeing what he went through also really inspired me to try to work on dementia and Alzheimer's disease and why the brain degenerates with time. So that's just a little bit about my background. And now we get to the talk, about what I'll be talking about today. So first I'll just talk a little bit about what we know about Alzheimer's disease. Then I'll talk about the normal brain versus the diseased brain. In the introduction, she mentioned ApoE, which is the protein that I work on. So I'll talk a little bit about my research. It's scientific and a little bit hard to understand maybe, but if anyone has any questions, please stop me and I can explain further. But I'll do my best to explain a bit more about this protein, talk about my research and some of the other research that's going on, and then finally some of the few drugs that are currently available, and then what we can do to prevent Alzheimer's, which is actually not very much at the current time. So, on to Alzheimer's disease. So as already been mentioned, it's the most common and devastating form of dementia. It affects currently 5.3 million Americans, and this number will double in 10 years, and then more than triple to 15 million people by 2015. The impact on families cannot be measured in dollars, but currently, well, in the year 2010, it was estimated to be $172 billion that was spent in the care of Alzheimer's disease patients, but not only in their care, but also in the loss of workforce because of family members having to take care of their loved ones. And this cost is then expected to increase by 85% by the year 2030. And presently, there is no effective medication that will cure, slow down, or reverse the cognitive decline that we see in Alzheimer's disease. So some of the characteristics of this disease: it's progressive, it's a degenerative brain disease with a gradual onset, and it's characterized by a loss of memory. So what is causing this is an atrophy or loss of brain cells in specific parts of the brain, and I'll talk about this a little bit more. And they're called hallmarks of the disease. This is what you see when you want to say for sure that the type of dementia that this person has is actually Alzheimer's disease. And these are called plaques and tangles. And there are different proteins that cause these problems. The ones that cause plaques are called amyloid, and the ones that cause tangle is a protein called tau. And then finally, another thing that happens is a depletion of neurotransmitters. So when two nerve cells talk to each other, they release little chemicals called neurotransmitter. And these are also depleted in this disease. So what about the healthy brain? So your brain is the most powerful, is it off the slide a little bit? Yeah, it's fine. So your brain is your most powerful and complex organ in the body. So it controls your emotions, it helps you to think, it helps you to feel. So basically you can say that your brain is you, it's your essence. So the brain is actually divided into lots of different parts. You have two hemispheres, and then within each hemisphere, the brain is divided into different lobes. And each of these lobes have a different function. So while I was going to school, we learned about this guy. So he was actually alive while I was studying, and to protect his identity, he was simply referred to as patient HM. And he was a really important patient because when he was, so this is better. The P's are coming out a little harsh. He actually had a bicycle accident when he was about 9, and after this he suffered from epilepsy. It was pretty bad. So he was finally referred to a neurosurgeon to help to cure him. And at the time, this was in 1953, the surgery, it was suggested to cure him because it was so bad that he should have surgery. So they went in and took out the part of the brain that was causing the epilepsy. Now, this cured the epilepsy, but after the surgery, he was never able to form another new memory. So he lived for 55 years after the surgery, and he could never remember something new. So you would walk into a room and you say, "Hi, my name is Yaisa." You walk out, walk back in, he has no idea who you were for 55 years. So this was able to tell scientists that this part of the brain is pretty important. Maybe you shouldn't cut it out. But what was also very interesting is that he can form different kinds of memories. So for example, he worked with scientists for the duration of his life, for the 55 years after his surgery, and he contributed a lot to our knowledge. So he would, for example, learn to walk a maze. Now, he could never remember doing this maze, but with time, he got better at doing it. So it was his working memory. You know, it's just simply by doing something over and over, you can get better at doing it, and that's not a conscious thing. So that memory was fine, but remembering having done the maze, he could never do. He could remember everything that happened before the surgery, but could never form a new memory after. So this really led to a lot of new information about the place that memories originate, and that there are different types of memory, and not all of them originate in the same place. And actually you can see here how his brain looked. So this is a normal brain, and then this is the part of the brain that they cut out from his surgery. So you can see here. So this part of the brain is called the hippocampus. And this is the part of the brain that degenerates first in Alzheimer's disease, which is why we see memory loss. So again, if you look at a normal brain here, this is how it looks. And if you take a slice out of it like this, you take a slice out and lift it out, it would look like this. And then if we look at the hippocampus again, which is this part for memory, it's these little parts down here. When we look at a brain from an Alzheimer's disease patient, you can see that there's a lot of loss. It's a lot shrunken. You can see there are spaces in between where there shouldn't be. And then again, if you take a slice out and look at it, you can see the difference between a normal brain and an Alzheimer's disease brain. And again, looking at the hippocampus, you can see the incredible loss that you see here, which is why we have a lot of memory loss in this disease. So what is actually being lost in the brain? So the brain is made up of nerve cells, and these cells all talk to each other. So you have, the cell looks like this, it has a cell body, and it has these long arms, and they reach out and talk to other brain cells. And they also, for example, one would originate here and go all the way down to your toe, for example. So those are these long axons that go. So what starts happening is first these little chemicals that I said, where these two cells talk to each other, These are called neurotransmitters, and these are depleted, so they don't work anymore properly. Then you start losing the connections themselves. So one cell is here, the other is here. They would usually talk, and then that's gone. And then finally, the cell itself dies, and you have cell loss in the brain. So it's a gradual process. So again, this is just a little cartoon of how the cells talk to each other. So again, you have this cell body, and these arms going out and talking to other cells in the brain and making these connections. And then you have toxic proteins that build up slowly over time in the brain. You remember at the beginning of the talk I mentioned the tangles and the plaques? So this is what like a plaque would look like. So these happen outside of the cells, and they form these big plaques here that kind of get in the way and start disrupting the connections between the cells. What also happens is that you have tangles, and these happen inside of the cells and kind of kill the cells from the inside. And then the other protein that I want to talk about, which I'll focus more on today, is called ApoE. So just to give you some more background about ApoE. So with your genes, you get one set of genes from your mom, one set from your dad. And ApoE is actually a very common protein, all of us have it. And then there are two different types of the protein. There's the ApoE3, which is a good ApoE, which hopefully all of us in this room have. It's the most common form, and it's the so-called normal form. There's another type of ApoE called the ApoE4, which is the bad ApoE. This is the abnormal form, and this is associated with Alzheimer's disease. And unfortunately, this bad ApoE4 is actually not that rare. About 25% of the population has it. So if 4 of us are standing around, most likely one of us has this bad gene that's going to increase your risk of getting Alzheimer's disease. So just in the normal population, as I said, it's 25%. But if we take the Alzheimer's disease population and look at their genes, within that population, 65 to 80% of Alzheimer's disease patients, they have the bad form of ApoE. So it really is highly prevalent in the Alzheimer's disease population. So having this bad form of ApoE not only increases your risk of getting the disease, but it also decreases the age of onset. So if you don't have ApoE, because the biggest risk factor for getting Alzheimer's disease is simply living too long, basically. So age is the biggest risk factor. So the older you get, the likelihood that it may happen is there. However, if you and it may happen around 75 to 80 years. If you do have ApoE4, however, this age of onset decreases down to 65 to 68 years. So this is just a graph showing the risk of developing Alzheimer's disease around the age of 60. So again, as I said, if you have 2 E3s, which again, hopefully all of us have, there's still some risk of getting the disease. However, if you have 1 E4, so you get 1 E4 from one of your parents, if you're a man, this doubles your risk of getting it. If you're a woman, unfortunately, this almost quadruples your chance of getting Alzheimer's disease. If you have 2 E4s, so 1 from each parent, your risk as a man goes up 10-fold, and as a woman goes up 12-fold of developing Alzheimer's disease. So how might ApoE4, which is the bad E, adversely affect nerve cells or neurons in the brain? So if there's some kind of injury happening, and unfortunately, simply getting older is in some ways an insult to your brain because it's hanging around for quite some time, and so aging itself is causing some neuronal damage. Or if you have head trauma, which is what I'm working on now, so some kind of traumatic brain injury, so the soldiers, our athletes, our car accident victims, if you have head trauma, this also increases your risk dramatically of getting Alzheimer's disease. Or if you have poor oxygen delivery, so suffering from a stroke like my grandfather, this also increases your risk. So all of these things can cause neuronal damage. And usually, this protein ApoE, it's in your body for a good reason, usually. So it should be there to repair and to protect your nerve cells. So it's there to help when your nerve cells become damaged. But the ApoE4, because it's the abnormal form, it can't do its job properly. So there's poor repair, and there's no protection, and this is why we then have Alzheimer's disease, because it can't do the job that it's supposed to do. So one question that I get, at least a lot from my family, is how do you even study Alzheimer's disease as a researcher? Because we use animal models in the type of research that I do. We don't work with patients, it's not clinical work. So our model is the mouse. And then my younger sister was even like, how are you even going to test memory in a mouse? You can't ask it a question. You can't ask it where do you leave the keys, where do you park your car. But she's a little sarcastic too. So, but the thing is, although they're very tiny, they have brains just like we do. They're also mammals just like we are. And the setup of their brain is very similar to ours. So again, looking at this slice of a normal brain, do you remember this part of the brain, which is the Hippocampus? Excellent. Which is responsible for your memory. So a mouse brain has the same thing. So they also have a hippocampus. So it's in a slightly different place, and it's actually a little bit bigger in relation to the size of the mouse's brain. But they also form new memories. And we can use this to be able to test memory problems, for example, in a mouse. In a different way, obviously, than a human, but we can use these as a model. So what we do to test these mice, to test for memory problems, it's called a water maze. So most people, when they think of testing mice in a maze, they kind of see this kind of grid with places to run and maybe a piece of cheese somewhere that the mouse is going to find. So that's not exactly how we do it. We do it in something called a Morris water maze. So it looks like this. So it's a tank of water, and you put the mouse in. So mice are really good swimmers, but they don't particularly like it. So they're very motivated to swim really quickly and try to find a way out. And we give them a hidden platform, so they learn to swim, and over time, find this platform so that you take them out. And then we can use this to test problems. So, for example, at the beginning of learning these tests, you put the mouse in, and of course he has no idea what you want. He was in his cage, happy with his roommate, and then suddenly you take him out and drop him in some water. And they kind of freak out a little bit, it's like, oh my God, what's happening? And they swim around frantically. But then you guide them on the first day to say, hey, there's this hidden platform, and if you sit here, I will take you out. And then as time goes by, within 3 days, you put them in and they swim directly there, sit down, and it's like, hey, take me out. So you can use this test. So the other thing that we are able to do in research is introduce human proteins into mice. So as I said, in humans, we have these genes called ApoE4 or ApoE3, which is a good one. And we can introduce these proteins into mice so that the mice now have the human protein. And the protein also behaves the same way in the mouse like it would in the human. So what we saw when we put these proteins into the mice is that mice expressing the ApoE4, which is the bad E, when they had it in their brains, just like humans, they had a loss of nerve connections, they had dying neurons, and they also had impaired learning and memory. So again, this is how this water maze test looks. So you have this tank of water, you drop the mouse in, you are kind of hidden off to the side with your computer, and then there are cues around and a camera measuring what the mice does. So I actually have here a little video. So this is my research associate here. She's putting a white powder into here, it's not toxic, so that the mouse can't see the platform. And then this is where the hidden platform here is located. So if we just put a regular wild-type mouse, he's not manipulated in any way, and you put him in, with time you can see he can swim really well, and he finds this hidden platform and sits down, and he's like, get me out, right now. We take him out, and we put him back into his cage. This guy here is an ApoE3 mouse. Remember, ApoE3 was the good ApoE. So this mouse also really learned to locate that platform, and they do so by using cues, looking around, and they can figure out where it is. And they swim directly, and they sit down, and you take them out. This guy here is a mouse with ApoE4. He also had the same amount of training, but he has no idea where this platform is. It's like parking your car and you can't remember. And you're up and down in the park house looking for the car. And that's what's happening with this guy. We only let them swim for 1 minute, so he's not in there forever, swimming around. But you can see, he can't remember where it is. Although he was trained over 5 days, he still can't remember where the platform was located. And then we can now use these animal models to test different drugs. So for example, we can try to give this mouse a drug to try to rescue this learning and memory problem that they have. And this is how we do our research. So again, how might this bad ApoE4 be causing these neuropathological effects, the loss of connections, the loss of cells, and also the learning and memory? So one of the major clues that we have is that the structure of ApoE4 is different from ApoE3. ApoE4 has a structure that leads to pathology. So this is, it's a little bit complicated, but the details are not important. It's just that you can see here, ApoE3, it kind of looks like an open claw like this. And ApoE4, it looks like this. This protein can do its job properly. So if a nerve cell is damaged, this protein can go in and help to repair it. This one, because its structure is not as it should be, it can't do its job properly. So a lot of the work that I'm currently doing is trying to find a small molecule which can hopefully in the future be used as a drug that converts ApoE4, which is the bad E, to an ApoE3-like molecule, and this then abolishes the detrimental effects of the pathology that we see. So this is how it would look. The ApoE4 is closed. We would hopefully get a drug that would go in and simply slip in between and open it so that it's more ApoE3-like and has the open conformation. Okay, so how do other Alzheimer's, so that was just a little bit about my research. So just some general talk now about some other Alzheimer's disease drugs and how they work. So remember I said the nerve cells, they talk to each other at these areas called synapses, and you have a loss of these neurotransmitter, a loss of these connections or synapses, and then a loss of cells. And then you remember I mentioned these different proteins called amyloid here, which form these plaques outside, and then tau, which form the plaques inside. So if we actually take a slice of a human brain and then look at the pathology from an Alzheimer's disease patient, this is how this brain would look. So a regular human brain, if you were to do the same kind of staining, it would be clear, it would be clean, and you would see nothing. From an Alzheimer's disease patient, you see these kind of sticky, junky looking things here. These are the plaques which happen outside of the cells. And if you look within the neurons itself, this is a nerve cell, this should also be clear. But because they have these tangles inside, you see it's lighting up here. So these are the tangles that kill the cell from the inside. So currently with the drugs that are available, they're not that many. These are the FDA approved drugs currently. There are 4 here, and these all act more or less in the same way. So they inhibit the breakdown of one of these chemicals, and as I said, they talk to each other using these chemicals. It's called acetylcholine. So, and this is a neurotransmitter important for cognitive function, and these drugs here help to keep that neurotransmitter around. And this has been shown to increase cognition in mild to moderate Alzheimer's disease patients. And then there's another one called lamentine. This acts on another neurotransmitter called glutamate, and this also has been shown to improve cognitive function and social and motor impairment a little bit. But although there are 5 drugs, none of them work very well. None of them slow the disease, none of them reverse the disease, and there's certainly no cure. And then here, this is actually a very depressing slide. It looks a little bit complicated, but the take-home message from this is that there have been many, many, many clinical trials for Alzheimer's disease. And apart from the 4 that I mentioned, and lamentine, which would be the 5th one that goes in here, all of these studies have failed. So this is why we need research. We still need advocates, that we can have funding, because funding is gradually getting less and less and less for us to do the work that we want to do. We still need a lot of research to be able to find a cure, or find a drug that at least slows it down, or stops it, or hopefully cures it. But one thing that is also available is early identification. So I'm a little bit torn about this. I do think that knowing exactly what's happening and having some certainty that, okay, your family member or loved one has Alzheimer's disease, this can bring some relief for sure. But for me, or maybe for you, doing genetic testing and knowing, for example, that you're ApoE4 positive, in some ways, for example, maybe if you want to be a boxer, not so good if you have ApoE4. If you want to play football, maybe not the best idea. If you are an athlete, so in these situations, if you're a soldier, maybe it's positive to know your ApoE4 status, so it can help you make decisions long term. But as I said, because there's no drug and there's no cure, knowing that you have this kind of ticking time bomb present, I think it's a personal decision that everyone can make. So there's definitely genetic testing for not only ApoE, but there are also other genetic markers for Alzheimer's disease. There are also brain scans, and these can show these plaques that I showed in the brain. If you do a brain scan, they can show up that you have these plaques present. There's also cerebral spinal fluid, so you do a spinal tap, and they can check within your cerebral spinal fluid to see if there are certain proteins that are present. And then there are also different biomarkers that different scientists are working on to try to be able to, with a relatively quick test, so just getting some blood and some plasma, to be able to identify that, okay, you have this protein increasing, so maybe your chance of getting Alzheimer's is present. So what can you do to prevent getting Alzheimer's disease, or to at least reduce your risk? being heart healthy. So anything that's good for your heart is good for your brain. You know, exercising, of course, there's been quite a lot of research that showed that exercising increases neurogenesis. So even while I was in school, you learned that you have a certain number of brain cells and that's it. And even my mom would say, you know, I would climb trees with my brother when I was younger. If you fall out that tree and you hit your head and you lose some brain cells, that's it. You don't have any more coming back. But this is not exactly true. We do have some neurogenesis that happens in the brain as adults. It's not very much, but it also increases with exercise. So exercising is definitely very good for your brain. Avoiding cardiovascular disease, avoiding obesity and diabetes. Also, maintaining brain stimulation, increasing education, learning a new language, etc., and having some novelty. And then also with your nutrition, you know, so having a balanced diet, getting all of your vitamins and your antioxidants and omega-3s through a balanced diet. And then finally, maintaining a good social and emotional well-being, because often what happens with retirement, and I also saw this happen with my father, you know, he had this big network while he was still working. But once he retired, this really dramatically decreased. You know, so you have to try to maintain contacts with your family. Hopefully you have a nice family and you get along well with them, and maintain those close contacts, you know, maintain going out with friends. And then, of course, there's been some research that showed that wine helped. You know, so you fund that one. So you don't want to have too much wine. Obviously that leads to other problems. You know, but having a glass of red, it probably won't hurt. Okay? So that's the end of my talk for today. So I want to thank the Alzheimer's Association again. Thank you for having me here to give this talk. I'd like to thank the administrative staff, the advocates, all of the scientists, and all of you also, the caregivers, because it does take a village to finally cure this really terrible disease. So I'm open for any questions.
Can you tell us about any of the drugs that you're using with mice that show some promise as far as reversing some of the effects of Alzheimer's disease?
So that's a really good question. So all of these drugs show promise in mice. Oh, I'm sorry. So the question was, can I talk about any of the drugs that we've used in mice that can cure Alzheimer's disease, basically in the mouse? And the answer is that all of these drugs have cured Alzheimer's in mice. So we have no problem curing mice from Alzheimer's disease. The problem is when we get to humans. Because the mice, they're genetically very similar to each other. And when we introduce the proteins, often we introduce them in isolation. So I study ApoE4, so we put that protein in. Or someone else is studying tau, you put that protein in. We even have some models where you put multiple human genes in. But within the mouse, it doesn't have the diversity that we have. Because even though you and I may have had a similar upbringing and we had a relatively similar diet, my genetic background is still very different to yours. So when you get to the humans, it's a much more complicated scenario that you have going on. So although, you know, using SP233, we put that in the mouse, we can cure the mouse. That mouse, you put it in the water, it finds that platform, and it's like, great. So we take that, we're very excited, we go to clinical trials, and then it fails. Another reason why some of these trials have failed is because often Alzheimer's disease patients are grouped as one group of patients. But you see here what I talked about with ApoE3 and ApoE4. So someone who has ApoE4 often responds differently to a drug than someone who does not have ApoE4. So that's happening now where they are breaking apart the groups of Alzheimer's disease patients into different subgroups and then trying the drugs again to see if it has a different effect in one population versus the other. But to answer your question, yes, we've cured Alzheimer's disease many times in mice. But unfortunately, no success so far in humans. So someone's coming with the mic. I think it's a little bit easier.
Could you comment on any kind of research or how this may apply to FTD and semantic dementia?
So FTD is called frontotemporal dementia. So it's different from Alzheimer's disease. I mean, the commonality is dementia, of course, but it's under a slightly different umbrella. So FTD, the main protein that's causing that problem is called tau. So this is the one that I said that causes the tangles inside of the cells. So in blue here, a lot of these are the tau drugs that have been tried. And so it's a different clinical trial that they would do for the FTD patients. And it's not my area of expertise. I work specifically on Alzheimer's disease. But some of the pathological cascades or pathways that we see are the same because one of the proteins we have in common, tau, it's both in Alzheimer's disease and in FTD. But also in that case, we don't have a drug. Yeah.
Is there any theory that explains the accumulation of plaque in Alzheimer's disease patients who do not have APOE4 genes?
Ask me the question again.
Is there any explanation of people who are Alzheimer's patients who have plaque who do not have the APOE4 gene?
Okay, so the plaques are caused by amyloid beta. It's a separate protein. It's APP, and this is cleaved and cut into little fragments, and this causes the plaques. APOE4, actually, one of its roles is not only to help repair the neurons, but also clears away these plaques. So an APOE3 an APOE3 person, their brain is better able to clear away these plaques and the amyloid that's building up in the brain. Whereas an APOE4 person, because again the protein doesn't look like it should, it can't clear away the debris and the plaques as well. However, there have been scans of older patients or people who have a lot of these plaques in their brain, so it's full of it, and they're actually just fine. So the plaques themselves are not necessarily correlated with having disease, because there are some people who have a brain full of plaques, and cognitively they're totally okay.
But what about the people who do have Alzheimer's and who do have plaque, but don't have the APOE4, don't fall into that percentage category that don't have the APOE4? Is there any theories about that?
I mean, so they're different within the APP, which is the one that causes the plaques. There are different mutations that can happen in that protein, such as presenilin and so on, and this is definitely much more aggressive, so it causes early onset Alzheimer's disease, so even in your 30s or 40s. And that's completely separate from the pathology we see with the APOE4. So if you have a mutation in that protein, you can have too much of this protein being produced, and this builds up in the brain, and this can cause pathology. But the two proteins, although they do interact, as I said, the ApoE is present, and it can help to clear away this protein from the brain. Even if you have a lot of the protein, it doesn't necessarily mean you will get the disease. Or you can be one of those people where you have a lot of the protein and you have the disease. Because kind of coming back to this question that was posed here, the human brain it's very diverse and reacts very differently. So my brain, for some reason, can handle a really high plaque load. And I'm totally fine. And my sister has far less plaques in her brain, and she has Alzheimer's disease. So it depends on the individual. Right?
Yeah, there's, excuse me. I believe there'd be non-clinical information out there about coconut oil relating to positive benefits. Can you comment on that?
So I had that question posed to me before. As a scientist, we like to do studies, and you repeat that study at least 3 times. Hopefully someone else can do the same study and see the same thing before we believe that what we're seeing is true. And then often, you have this control, it's peer-reviewed, people look at your work, make sure it's solid before it's taken into clinical studies. There's definitely anecdotal evidence that has showed that coconut oil has helped. That's a very small number. As a scientist, I would say that's not very strong information, and I think that's also a personal call. But I know this question was posed before at the Alzheimer's Association, and Elizabeth, you had a little bit of an answer that you wanted to give towards that, because I know you get a lot of information.
We get this question all the time. I talk about coconut oil a lot, and what Yaisa said is true. It's really the reason it became a topic is because a neurologist whose husband had Alzheimer's started thinking through coconut oil and how it's structured and thought, well, maybe this will be helpful for him, and what the heck, let's try it, right? After, with Dr. Zwilling Andrews was talking about, it's not like Aricept is going to bring you back to where you were, so maybe coconut oil will be helpful. And she believed and showed some evidence that in her husband it was helpful. Unfortunately, there haven't been studies. As Yaisa said, there may be some small related things here or there, but it's not clear cut. So for those of you who have heard about it and are trying to figure out, well, what do I do here? There's not scientific evidence that's going to tell you this is helpful. All right? It's just not available. However, with anything related to your health, don't you do kind of a cost-benefit analysis and you think, well, how dangerous is coconut oil to take? How expensive is it? And what other side effects might there be if my loved one takes coconut oil? So this is what you have to do. I know a lot of people who said, let's try it. Let's see. I mean, the risks are probably pretty low, and if it helps somebody do better, maybe it's worth it. But that's a very personal decision. So what's hard for us, similar to what Yaisa said, is we do go for the research. Oh, I'm sorry. We do go for what's the science telling us. And the answer is the science isn't telling us anything about this yet. There aren't large-scale studies to back you up. But it might be something that you would want to try. If you do, let us know. Let us know if it's helpful. I know people on our website have been commenting on this. Some swear by it, and some say it doesn't help. I want to say one other thing, though. I had a woman tell me that she was thinking of taking her dad out of a nursing home because the nursing home wouldn't give him coconut oil, and she thought if she brought him home and gave him coconut oil, it would cure him. I do not believe that it's likely to cure Alzheimer's, so I cautioned her. I said, it's not a miracle substance, and so just be careful. Don't make any rash decisions around hope about coconut oil. So, coconut oil is free. Yes.
Yes, I'm interested in how much genetically you're looking at metabolism related to these genes in other areas and nutrition. For example, people who have genetic markers for breast disease, diet makes a difference. People, back when I was in nursing school, way back then, people had heart disease, but nobody thought diet, exercise, or anything made a difference. So, like you said, these genetic markers even break down smaller, and I'm wondering, it's like when you bake a cake, all you have to do is leave out one ingredient and you don't get a cake, you get something else. So all those things have to work together, and I'm wondering, I know you discussed nutrition in that little framework there, but as far as the genetic marker, do they process, liver function, demethylation? Are you looking at other processes in the body and supports that would make a difference?
So that's a really excellent question. So again, coming back to ApoE, which is my area of expertise, this protein was first discovered actually as a heart protein. So it's actually a lipid transporter, so it transports cholesterol, which is how it helps to fix the neurons. So my current boss, Dr. Maley, he spent his life working on ApoE. So it was a heart protein, and it has a role in cardiovascular function. It's also made in the liver. And then years later, they found it in the brain, and they were a bit confused because what is a heart protein doing in the brain? And then they found that it was associated with Alzheimer's disease. So to kind of bring the two together, because this protein, its main role is actually to transport lipids and fats, and it has a role in cardiovascular disease, you can see that the two actually converge. So if you have a poor diet and you do have cardiovascular disease, this is also in the same feedback loop that's with your brain, because the same protein is in your brain not doing a great job, is in your periphery, in your heart not doing a good job. So you can imagine where they collide. So just in general, having a good diet and avoiding cardiovascular disease is a good thing to do. And also there's been a lot of work, a lot of work looking at omega-3s and that in cognition, and that has been shown to have a positive effect. But bringing it back to ApoE, that protein is also involved in heart disease. So the two definitely converge with metabolism and also with Alzheimer's disease. And again, ApoE4, because as I said, it increases your risk of getting Alzheimer's. So we often talk about now secondary hits, because unfortunately, if you have E4, you already have a first hit, and it's working against you. So if you have this protein, having any secondary hit really increases your risk of getting Alzheimer's disease. So if you smoke and you have E4, that's really bad. If you have E4 and you have cardiovascular disease, if you have E4 and you have a traumatic brain injury, if you have E4 and anything else, it really then sends up your risk more than tenfold of getting the disease.
Yes, hi. I'm just curious about a couple of things. Number one, could you give us some idea of the scope of research that is going on today, both nationally and internationally, to deal with Alzheimer's? And secondly, what role does the federal government play today in funding research? Is it mostly private? It sounds like your institute is a private organization. I'd be interested in knowing where your funding comes from, and then do you receive any kind of federal assistance at all? Thank you.
So the scope. So there's a conference called Society for Neuroscience, and it just happened 2 weeks ago in New Orleans. So it was a great place to go for a conference. And there were 30,000 neuroscientists there, and a pretty huge chunk of them are working on Alzheimer's disease. So I have friends in Germany, in Japan, in Singapore, so there are a lot of people working on this disease. With respect to funding, now is not a great time for neuroscientists. The disease is very complicated. You know, it's not like, let's say, cancer. It's also a very important disease. Obviously, many people suffer, but to study cancer, so cancer is basically a cell that rapidly multiplies and takes up blood resources and space in your body, and eventually, of course, can lead to death. Now, if you want to study this disease as a scientist, you can simply study those cells in a dish and try different drugs to see if you can slow down the growth of these cells, for example. With Alzheimer's disease, there is no easy way to study this disease. We have to go to animal models. Trying to maintain a colony of thousands of mice, which is what we have, it's very expensive. And also from what you've seen, even though we have these mice and we do all of this work and we cure Alzheimer's disease in mice, it's not translating to the clinic. So as a result, many companies have shut down their neuroscience departments because there's no drug. And I mean, although within companies, their main goal also is to find a drug and to cure, they also have to look at their bottom line, yeah? And they're not making any money, and these programs are very expensive. So with respect to funding from the federal government, this also under the Bush administration went down dramatically. It has gotten a little bit better under the current administration, but not great. So the number of grants that are available have also decreased significantly over time. It's getting harder and harder to get grants. Whereas before, if you're in the top 10%, you have a really good chance of getting a grant. Now you have to be in the top 2% to get a grant. As a new researcher coming out and trying to get funding, it's almost impossible. You know, I have a lot of friends who are looking for jobs and can't get right now, and it's a little bit heartbreaking for us. I mean, we studied for a long time. And then I just heard there's a company who, it's like a temp agency for PhDs, where we're rented out to do research. And this, I mean, I almost cried when I heard it, to think that you work so long and study this hard and then you can't get a job. And it's because the funding is drying up. So the Alzheimer's Association also provides some funding, which is really great. I've also gotten a couple awards from them, which has helped with my research. But again, there, I mean, you also spoke about the applying for federal funds and so on. So that has increased compared to previous years, but it's nowhere near what we need to be able to do the work that we do. So with respect to money, it's not a great situation right now. So as a result, we also go to a lot of private people. Because it's such a prevalent disease, many people are scared. And we have some very rich people who are scared. So we go to them. Yeah? And we say, you know, the work that we're doing may not be able to help you directly, because we're still, from that graph that I showed, we're far away from having a wonder drug that's going to work. But we may be able to help your grandchildren, your children, and we'll definitely be able to help your great-grandchildren. So that's where it is right now. So we go to, I do a lot of fundraising, and I go out and I try to talk to private donors. So it's a combination of private money and money from the federal government that we have right now. And from the Alzheimer's Association. Any more questions? Yes? Yes.
Hi. Since Alzheimer's, the curing of Alzheimer's doesn't translate to humans,
Yeah.
why are people going to continue with the mice? Is there an idea of making the research a little bit more complex to address the non-translation and all the factors, or is somebody thinking about a different way of doing research?
So that's an excellent question. So you guys heard her term mouse. Yeah, we've cured that one quite a few times. So it's a really good question. These mice are genetic models often that recapitulate a particular aspect of the disease. So as I said, our mice have ApoE4. Someone else's mice or mouse line would have tau, which also causes FTD, which was your question, for example. And someone else would have an amyloid model. And we do work in these different areas. So what is happening now is that we're crossing these different lines, mating them with each other. So that one particular mouse has 3 different things that bring together all of the different proteins that play a role in Alzheimer's disease. So that's one direction that it's going in. Another is that the majority of the work that has been done in the last 2 decades have focused on amyloid, which is the one that's causing these plaques. The field is very focused on this particular protein because it's the one where you see the plaques, and many of the drug companies, their research is still focused on amyloid. That has been changing. I'm not sure if you guys heard about bapineuzumab. Yeah? So it's another clinical study that actually failed. Can I actually just come down here for a second? So this clinical study was, yeah. So this one made the news. It was pretty big. And it was also focused on amyloid, again, this protein. So what they saw, this one was an antibody that was given that is shown to pull these plaques out of the brain. And actually, one person who took part in this study was a neuroscientist from UCSF who developed this drug years ago in her career, and she now has Alzheimer's disease. So she was a part of her own drug study, and the study failed. So what is happening now is that the field has to do some soul searching about what we think is actually causing Alzheimer's disease. Yes, amyloid plays a big role. It's present in the brain. You have these plaques. If you have a mutation in the protein, it definitely causes the disease to be more aggressive and earlier. But focusing on this protein to cure the disease may not be the direction to go in. So that's what's happening now. There's a bit more focus on tau, which is the FTD protein. There's a bit more focus on ApoE, and looking at other avenues that we can attack the problem from, in addition to amyloid. Another thing that came out of this study is that, again, in the beginning, they treated all of the patients as one group of Alzheimer's disease patients. But when they went back, they then separated them into ApoE3 and ApoE4 because the 2 groups respond differently to drugs. So this is another thing that has come out of mouse studies, that the protein acts differently in humans and also in mice. So we do have to treat these 2 groups separately. And so that's where it is right now. Going to more complex organisms to study the disease, we can genetically modify more complex organisms. It's also very expensive, for example, if you get to monkeys, where some research is also done. You can imagine the ethical concerns that are surrounded. Even using mice, obviously we have lots of protocols to treat the mice ethically and responsibly. And going to monkeys, this just goes up exponentially. And the cost of keeping, we have maybe 8,000, no more, maybe 20,000 mice. Monkeys, the space that you need to keep even 100. And the cost, and as we said, the funding is also going down, so yeah.
I wasn't talking about going to a more complex mammal per se, but a more complex research project that as you mentioned, kind of
that combines the 3, yes.
Things, because maybe you can't just separate out each different little thing.
Yeah, yeah. So that's where it's definitely going, crossing these mice and having the different proteins present in one organism and trying to tackle it from there. That's happening right now. It's a good question.
Yes, I would like to ask, you say you do research in different countries.
Yeah.
So what is the prevalence of the disease, or what's the difference? Is there a difference with Alzheimer's in, say, Germany, the population of people in Germany, or here, or Africa, different places? Is it different, or is it about the same?
So I don't have a slide with me on the statistics. This is a problem that's happening worldwide. It's definitely increasing dramatically in Asia as their population ages, and they have, a very large population. In Germany, where I also studied, it's definitely a major problem there because they have more of a social democracy, where traditionally young people have put money into, you pay really high taxes there, but then you have a fantastic healthcare system, etc. But to maintain this, you have to have a lot of young people putting money into the system to support the population as it ages. And they have a bit of a pyramid now where you have a lot of older people And the Germans are not having enough babies, which is why I went in and I had one. But their population is aging, and they don't have as many young people to support the system. So it's a huge problem there also as their population ages and they have Alzheimer's disease. So it's a problem that's happening worldwide. In some ways, was traditionally more of a first world problem because in the third world where I'm from, you have other diseases that take you out before Alzheimer's can get you. But as the world health in general increases and the standard of living, people are living longer and longer everywhere. So, and as the population ages, as I said, age is the major genetic risk, the major risk factor for getting Alzheimer's disease. So it's a problem worldwide. Sorry, so would you like to say anything about that? Do you guys have any statistics apart from the US, like I had on my slide? Would you guys like to say anything to that? Okay.
So is the disease itself the same, just about the same with people?
Yes, it is the same. I mean, in first world countries, and for example within the US, you have a bit more of a comorbidity with obesity and cardiovascular disease because of our Western diet. Within Japan, they had a bit of a lower prevalence of it because of their diet with high fish and so on. But the disease itself, it's the same everywhere.
I can shout. Thank you. Guys, could you say a few things? I just came back from a meeting where I heard people talk about solanezumab being, sort of dissecting the data after the trial had failed. There were still some positive outcomes that one of the NIA groups identified, and I understand it's one of the drugs that's going to be used in the Colombian study with the people who have the familial form of Alzheimer's. And then also there is the dominantly inherited Alzheimer's network, which the Alzheimer's Association is helping to fund. And they're going to be using it in that group. So one of the problems for these drugs, as you know, at least one of the suggestions is that phase 3 trials have failed perhaps because we mount the trials with people who are really sick. We trying to do heart disease studies with people after they've had heart attacks or strokes. Talk a little bit about that approach to introducing some of these compounds at a different stage of the condition.
Okay. So in the case of the Colombian group, they have a particular genetic risk factor, which is separate to ApoE, which also predisposes them to getting Alzheimer's disease. So within that population, the plan is then to use this drug kind of prophylactically to prevent the disease. In that case, I think it's a great idea because this population kind of knows that the disease is coming with relative certainty. I think part of the problem with the research that we do would be getting healthy individuals who have no cognitive decline, not even MCI, because some of the studies start with MCI, which is mild cognitive impairment, before it starts getting really bad. But even even within that group, the MCI patients, the cascade is probably so far gone in the brain before you start showing problems that by the time you start giving the drug, even if it's relatively early in the physical or clinical manifestation of the disease, for your brain, it's already past a certain point of being able to be helped. The problem with going before that point would be convincing someone to take a drug when you're completely healthy. So what's the likelihood of everyone in this room taking this drug tomorrow, knowing that there's a possibility that you will get Alzheimer's, so you should take this for who knows how many years to possibly prevent it? How many of you would sign up for that clinical trial? A couple. And a couple maybe because you have a family member and you're concerned, but within the general population, it's really hard. And that kind of comes back to the other question of how do we actually tackle this disease, because how do we decide when is the right point to start giving treatment? How do we convince healthy individuals to take a drug? Is it that we do genetic screening and all of the ApoE4 people potentially take the drug? That could be one way to go because your chance of getting it is pretty high, especially if you have 2 E4s. So as he said, there have definitely been some positives that have come out of these studies where we know if you give it to, for example, people with another genetic risk factor, it's possible it can help in that population. But the vast majority of people who get Alzheimer's disease do not have these aggressive mutations. Many of us will have the APOE4, which increases your risk, but it's not very aggressive. You still get it relatively late. And then again, by the time you start showing symptoms and you're in a clinical trial and you get the drug, it's not working. So it's definitely a problem that we have of how early do we test, how early do we treat, how early do we try to intervene. Many drugs also have side effects, you know, because we are modulating a protein that we know can go wrong, but if you start tweaking with this protein before it's going bad, what happens then, you know? So it's definitely a problem that we have. I'm sorry I don't have more good news.
Could you say the name on top of the screen there? Pronounce that.
Bapineuzumab. Bapineuzumab. My pronunciation is sometimes different from Americans though, is that right? Bapineuzumab. Any other questions? Yes. Mike, or do you want to shout?
If they're prescribing all these statin drugs for lowering the plaque in our cardiovascular system, is there any indication that there might be a side benefit and is it a similar plaque that is being built up in the brain?
So with the statins, these are cholesterol drugs. And these drugs were, of course, initially given because of heart problems and too high cholesterol. And what they saw kind of retroactively when they followed these patients who were getting the statins was that they had a far fewer incidence of Alzheimer's disease. So it was like, oh, we were using this drug for one thing, but maybe it can help with something else. So yes, it does work to some extent. But again, those people were taking that drug for a separate problem early in life. And it has been shown to help. But it kind of comes back around to the same question that we had previously of if you don't have cardiovascular disease, you have no symptoms of Alzheimer's disease, how willing are you to take statins in your 20s, for example, and take it for the rest of your life? It's possible that it will help, but there are definitely issues with treating healthy persons, not patients, with a drug long term when we don't know the effect of that drug long term, especially in someone who's healthy, you know, and it does treat the same plaques that we see, so it does work in Alzheimer's disease. But it hasn't been shown to be a cure.
Can you speak to where we are with pre-symptomatic diagnosis?
Where we are with pre-symptomatic diagnosis, so so that kind of, comes back to here. So, well here. So, you can do genetic testing if you have no symptoms, but you have a family member, or 2 or 3, who have gotten Alzheimer's disease. It's possible that you have APOE4 in your family. So you can definitely go in, it's a very easy test, you can get it from your saliva, just a swab, or a blood sample, and I can test you in the lab and I can tell you if you're E3 or E4. So in that sense, yes, this is pre-symptomatic. You will simply know if you have the disease, if you have the gene. But the gene just increases your risk. It's not 100% sure you're going to get it. You're 4 times more sure to get it. If you have 2 E4s, you're 12 times more sure to get it than someone else, but it's not for sure. So it's not a death sentence, let's say, if you have E4, so you're going to get Alzheimer's for sure. So that's one way. Again, getting cerebrospinal fluid, a spinal tap, and then you can check for these different proteins like tau, like amyloid, to see if it's there. Having brain scans done, this is obviously very expensive. So you can do all of these things, and there's some work going on biomarkers. I remember you spoke to that. Would you like to say something a little bit about the biomarkers? Huh? So there are definitely things you can do before you have symptoms to find out if you have a genetic predisposition or if you have some modulation and changes in your brain. But again, it's because we don't have a drug available, I'm not sure if that knowledge and the stress of that knowledge is healthy or good for you.
So the idea of biomarkers is, imagine if you could go in and get a blood test and find out if you have Alzheimer's or you're going to get Alzheimer's. And right now you can't do that. However, at Stanford, Tony Weisskore is a researcher at Stanford. He did a study on just that and found with tremendous resources, they can find a pattern of proteins in people's blood and potentially essentially assess the risk that that individual has, or even more so, looking at individuals with Alzheimer's, doing a blood test, and they can tell who has Alzheimer's versus who doesn't. So I was very excited about that. I thought, imagine getting a blood test in the process of getting a diagnosis where you knew, yes, you have Alzheimer's, or no, you don't. And I asked Dr. Weisskore, when would this test become available? And he said, oh, that test will never become available. And I thought, well, I don't understand. He said, the resources in our lab are tremendous. They wouldn't be able to do that commercially. So what we have to do is take what we've learned and then start to make it so that it's realistic, that it can be done in a commercial laboratory, and that's what they're working on. So we're not quite there, although all of these could be considered biomarkers. It's an independent marker. But right now, it's independent marker of your risk of developing Alzheimer's. So even if you get the spinal tap, it's not guaranteed that if you have, say, a positive test result that you will get Alzheimer's. It's close to that. We're getting to the point where that spinal tap looks like it's a very definitive marker, but it's not quite there. So that's what the field is looking towards. And it really is to look at drugs like bapineuzumab to say, if those of us in the room had positive biomarkers, so let's say half of us, God forbid, had positive biomarkers in this room, and we knew that 10 or 15 or 20 years from now we would get Alzheimer's, how many of us would volunteer for the bapineuzumab study? I think I would. If I don't think I'm going to sign up for that. But it makes a big difference, doesn't it? And that's why early identification is important, not so much for everyday living, but for research. Because if we can find people before changes take place, or really early, maybe we can stop it from progressing. And that would change the world. That would make it a different issue. It'd be, how do we get everybody tested, and how do we afford to get them treated? But that would be a good world to have to deal with those problems.
So that's really excellent. So that actually was the next point I was going to bring up, because I know, especially as family members of people caring for their loved ones and concerned about clinical trials and when or if one should get involved. And that's a really important topic that Elizabeth just brought up. If these biomarkers are available, or if you do think, because you have multiple family members, for example, who have had or passed away from Alzheimer's, maybe getting that genetic testing, finding out that you're E4, and possibly then applying for a clinical trial. Because maybe in that group, we would be able to do something. And then, as she said, you would be more motivated to get involved in a trial because you know the likelihood is higher, possibly, that you would get the disease. Yeah.
Hello?
Frank.
My wife takes both Aristat and Namenda. And since there's no proof that they're not doing any good, how long should she continue? I mean, we were told by our doctor, essentially, if we can afford it, we should continue it. But that's kind of the answer.
Yeah. So I think that's a personal decision that you make with your physician. But I know there's a session coming up soon which will talk about giving care. And those are people who definitely have more expertise than I do in the way you care for loved ones. Did you want to say something to that? Or,
yeah. Oh, sorry. I think that's the right answer. My mother-in-law lived to 99, and she took both of those drugs right up to the very end. And I think the issue is we don't have data about the alternative, right? We don't know what happens, and she could afford it. And my sister-in-law, who was the primary decision maker, said, "Oh, Mama can afford it. Let's just keep doing it." Because maybe it makes, she was in a long-term care facility for the last 3 years. Maybe it makes her a little more compliant, a little easier for staff to care for. We don't really know. We don't know. Had we not spent the money, had we not done it, maybe it would have been the same. We don't know.
Do you want to grab the mic behind you?
I just wanted to ask if there were any clusters, any environmental factors that contribute to clusters of Alzheimer's disease. Often, you know, the environment is considered quote-unquote a trigger or something that is genetically already within us. So, just your comments, please.
So again, scientifically proven that this particular toxin, or I know for a while there was like some anxiety over aluminum. That hasn't been scientifically proven. So I can't really say too much to that. It's not within my area of expertise, so I unfortunately can't say too much to it. And as far as I know, there's no clear research that has shown that a particular toxin or environmental factor can increase your risk of Alzheimer's disease. Yeah?
I'm sorry if I missed this in your presentation, but is there an estimate of the percentage of the population that has ApoE4?
So in the general population, it's 25%, so 1 in 4. And within the Alzheimer's disease population, it's 65 to 80% of them have E4. So it's enriched within the Alzheimer's group.
This is kind of not exactly a medical question, but if you do the genetic testing, and you may not be able to answer this, but perhaps somebody else in the room could, what impact does that have on your future insurance coverage?
That is an excellent question. So we, because of the, so I see Elizabeth jumped up, so I'm sure she's going to say something. Because I work specifically in ApoE in that area, and now looking also more towards traumatic brain injury. And for example, I have 2 sons, for example, and one is starting to get kind of interested in football, and there's no way he's going to play. I have to figure out a way to guide him, because if I just say no, obviously he won't do it more. But, so this is also something we've thought about, you know, should very young people be tested for ApoE? And how would that affect your potential coverage? Should you, if you have a family member, should you get tested, and how would that affect your coverage? But I will pass it over because I think she's like,
yeah,
a really good answer.
It's a good question, and it's a good thing to consider before you send off your cheek swab to find out whether you're ApoE4 positive, because a lot of times people will say to us, well, I want to get tested because I'm thinking of buying long-term care insurance.
Yes.
Right? I want to see if I want to spend the money. And the reality is, on the application, if you find out that you're E4 positive and you do the application, there will be a question related to that in some way. So either you will, and if you fill it in, say, yes, I have had genetic testing and these were the results, you will not be able to get that coverage. And if you say, no, I didn't have it, but in fact you did have it, then you're kind of lying on a legal document. So what we encourage people is first think very carefully if you really do want the results of the test, and think through all the ramifications of finding out your genetic makeup. And then secondly, if you decide, yes, after careful consideration, I would like it, and one of the perks might be deciding whether to get long-term care insurance, get the long-term care insurance first. Okay? And then if you find out that you don't have E4 and you want to cancel your policy for another 10 years, then go for it. But you don't want to put yourself in a position of feeling like you're doing it anonymously and under the table, and then you're in a position of lying. I think it's difficult. The other thing is, knowing that you're E4 positive does not mean you will get Alzheimer's. It means you will be in a higher risk group. And so if you're thinking of getting the genetic testing because you have family members with Alzheimer's, we already know that you're in a higher risk group. So knowing that you're E4 positive, what will you do with that information? It's just something you have to do some soul searching about, what will I do with this? And is it different knowing versus not? And some people will say, yes, it's different, I want to know. And a lot of us would say, I think maybe it's better that I don't know. But you have to make that decision for yourself before you do it, because it's not something to do on a whim. Sometimes people send off their genetic material for another reason entirely. They want to find out if they have a gene related to heart disease or cancer, and guess what they find in the mail? Then Alzheimer's gene that they didn't even know they might have. And so this genetic issue, I think, goes beyond Alzheimer's, but it's really important that you think about it carefully. So I think we have time for one more question, and then we're going to have Bill Fisher. Any other questions?
Okay, I'll end the questions with something way off the wall. I'm currently working with a group of middle school students who are involved in an after-school science program, and they're doing volunteer work at my day center, which primarily serves folks with Alzheimer's. And what they want to know is why, or is there research being done in nanotechnology in terms of little tiny robots going into the brain and cleaning out the stuff that causes Alzheimer's disease, doing repairs, and then everybody getting better.
Was my son in that group?
Yeah, these are 4th to 8th grade students.
Yeah. Because my son often asks different kinds of questions like that, like, well, you're a scientist. He asks like with this kind of challenging tone, you're a scientist, so why can't you just make a little robot? But there is nothing along those lines yet, so you have to tell your students, unfortunately, no. So this, what's his name? He just came to our institute to talk. So he works not with little robots, but with different types of, he was a chemical engineer first. I'm hoping his name comes back to me while I'm talking. Little things like this also worry me because of when you know too much, it's also not good. Like, I'm not going to stress. Okay, so he was a chemical engineer first, and he works with different types of molecules that can deliver drugs in a slow way, and different types of release, different types of polymers. So it's not a robot, but he definitely is working on different materials that you can deliver into the body that can have like a slow release of drugs and that can get through the blood-brain barrier and get into the brain. So there's research along those lines. But as far as I know, maybe in China somewhere they have little robots. As far as I know, there's nothing along those lines yet. So I think that was the last question. Thank you guys so much.